Let's continue our discussion with different parts and structures inside and outside the cell. Now remember, the cell has three main parts to it: the plasma membrane, the cytoplasm, and the nucleus. And if you want to call that plasma membrane a cell membrane or a phospholipid bilayer, whatever, there are several names for it. But going back to that cytoplasm, this is all the material that's outside of the nucleus but inside of that plasma membrane. So there's a lot of structures in there. There are different things in it, like the cytosol, which is the fluid portion of the cytoplasm. All living cells have got a certain amount of water inside of them and around them. There's the cytoskeleton, which we'll look at below a little bit sooner, a little bit later that is. Cytoplasmic inclusions, which are just collections of materials inside the cell, tiny little balls or vesicles of whatever that material may be. And then organelles, little structures inside the cell that each have their own little functions, sort of like tiny organs inside the cell itself. So there's our cytosol, the fluid portion. Remember, there's no such thing as pure water inside the body. There's always material in it, ions, proteins, whatever that may be. But coming back to the cytoskeleton here, you can see there's different parts and structures within it. The microtubules are good for internal scaffolding, giving the cell some shape and structure. They're also good for transport. You'll also see being used during cellular division. We'll look at mitosis further along. These microfilaments are also good for structure. They support these little external parts of the cell called microvilli. We'll look at further along. They're also good for contractility and moving things around the cell. Then these intermediate fibers are good for mechanical strength. Again, the cytoplasmic inclusions we mentioned are just collections of materials. Might be lipids, melanin, whatever, just depends on the cell. But going back to these organelles, think of these as like individual little workstations, sort of like how we have organs inside our body which have different functions. These organelles are different little structures inside the cell which have their own little functions too. Now the nucleus you always see inside of a cell, usually very large, usually singular, and usually towards the center of the cell. It has its own membrane. That little membrane is very similar to the cell membrane. We'll take a look at it on another picture further along. Has its own little double layer. There's a nucleoplasm, similar to the cytoplasm. This is what's largely the fluid material inside of the nucleus. You'll see this nucleolus, which is singular, or nucleoli, which is plural. Looks almost like a nucleus within a nucleus. And then again, that nuclear envelope, same as that little membrane there to the outside. Much of the DNA is located here. You say, well, where's the rest of it? The mitochondria do contain a small amount of the DNA, but most of it will be in this nucleus. Looking at chromosome structure, you always hear about chromosomes when you talk about the nucleus, this stored genetic material here. Well, when you look at it, it has a few different forms. Most of the time, the DNA is in the form of what's called chromatin. These are very thin, very tiny DNA strands. You can see they're complexed with proteins called histones. But during cellular division, mitosis we'll look at further along, this very thin strand chromatin condenses. So in other words, you pack it all together very tightly, and then it's called a chromosome. So it's all the same material. It can be loosely spread out in thin strands like the chromatin, are very tightly packed, as you see in the chromosomes. And you'll see these pairs of chromatids which make up the chromosomes, and each one of them is joined right in the center by a structure called a centromere. But let's look also at these ribosomes. Anytime you hear these ribosomes, you think protein synthesis. This is where the proteins are made. Now you'll see they have a small and large subunit on another picture further along, and there are a few different types. Ribosomes might be free, in other words, not attached to an endoplasmic reticulum. If they're free, they'll be making proteins, and those proteins are going to stay inside the cell for some function. But some ribosomes will be attached to a rough endoplasmic reticulum, still making proteins, but these proteins will be going out of the cell for some reason. Now look at that endoplasmic reticulum. Notice how there's a rough and a smooth. The rough ones look rough because they have ribosomes all over their surface. We'll look at a cell model of this further along. Again, anywhere you got ribosomes, you're talking about making proteins. This is where they'll be modified at this rough endoplasmic reticulum. The smooth endoplasmic reticulum looks smooth because you don't see the tiny little ribosomes all over the surface. And without ribosomes, they'll have nothing to do with protein production. The smooth endoplasmic reticulum is all about making lipids, detoxification, in calcium storage. And you'll see more of that calcium storage in them when you get to things like muscles and how they work. You'll also see within these endoplasmic reticulum the cisterna, interior spaces to the inside. We'll also mention the Golgi apparatus. This here is the modifying, packaging, and distributing center of the cell. So after one of these other structures, like a ribosome or whatever, has made its material, it'll often send it to the Golgi apparatus. Here it'll be modified, packaged up, and then distributed out of the cell. We got a little model shows that further along. If you look at what these Golgi apparatus look like, it looks almost like you stacked up a bunch of towels and mashed them in right in the very center. Vesicles are just little packages of some type of material inside the cell. Just by looking at them, you couldn't tell which one was which. You'd have to know something about what was in the inside or what was it formed. So there are vesicles called lysosomes, which contain digestive enzymes. So they're good for digesting, breaking down things. There are peroxisomes. These are a little bit smaller type of vesicle, and they contain enzymes which will break down fatty acids and amino acids. Hydrogen peroxide is a byproduct of this. You cannot let any acid build up in the body that will disrupt homeostasis quickly. There are also these proteasomes. Notice how these consist of large protein complexes. They include enzymes that break down and recycle proteins in the cell. Mitochondria. These are the power plants of the cell. Here's where almost all ATP is made. When you look at their structure, there's a cristae, all these interfoldings to the inside, and then the matrix, which is all the space in between them. But looking back at these mitochondria, these are your power plants of the cell. Remember, ATP is that energy currency of the cell. When cells need more energy, they'll make more mitochondria, and they get more active. And you'll see more about that when you get to hormones and such, like thyroid hormone. There are also centrioles and spindle fibers. These structures here are involved more with cellular division. So these centrioles here, you'll see, are located in the centrosome, which is a region close to the nucleus. You'll see that this is where microtubules form. They're very much involved with moving things around the cell. So that's where you see them used with cellular division. And you'll also see these centrioles divide at that time and spindle fibers forming all around them. Here are also some external structures you might see on the outside of a cell. Now, the smallest of them all is the microvilli. These are very tiny. You might see thousands of these on the surface of a cell if it has them. If an epithelial cell has microvilli, they are there to increase surface area. And you need to do that anytime the cell needs to bring in or put out. In other words, absorb or secrete a very large amount of materials. Then there's cilia. Now, these structures here are a bit larger and fewer in number than the microvilli. And they're always used for moving something over the surface of the cell. Look at something like your air passageways, your trachea and your bronchi and the upper parts of the respiratory tree. You'll see that these cilia are moving the mucus over their surface. You get all kinds of dirt and other material trapped in that mucus as you inhale air. This will move that mucus and get it up out of your lungs. Then there's the flagella. The only place you'll see this in the human body is the sperm cell. This is found on a cell. There'll only be one flagella, and that's it. Not large numbers like you might see with cilia and microvilli, just one flagella. And it's used for propulsion. It's like an outboard motor on the back of that cell. So if you look right here at this picture, this model, here to the very outside, the biggest cell membrane, plasma membrane, phospholipid bilayer, whatever you want to call it. Then you get all these organelles and structures. Here's your nucleus here, usually pretty large and takes up a lot of room inside the cell. You can see it has its own little doubled layered nuclear envelope or membrane, its little nuclear pores. And here you can see to the inside the chromotin, the little thin strands of DNA. And here are the little nucleoli. They look almost like a nucleus within a nucleus. Now looking at some of these structures on the outside, here you can see smooth endoplasmic reticulum in many places. The rough has ribosomes on the surface. Notice the smooth does not. So you can definitely see a difference in their structure when you look at them. Remember, the smooth endoplasmic reticulum is about producing lipids, detoxification, and storing calcium. The rough endoplasmic reticulum is about making proteins because it has the ribosomes and it's what they're for. And again, the ribosomes might be attached to the rough endoplasmic reticulum, or they might be free. That's what these little red dots are, free ribosomes. Again, free ribosomes make proteins that will be staying inside the cell. Those attached to the rough endoplasmic reticulum make proteins that will be going out of the cell. You can also see right here a Golgi body, also called a Golgi apparatus. After something like a rough or smooth endoplasmic reticulum makes something, it'll often send it to this Golgi apparatus. It'll be modified, packaged, and distributed out of the cell, and that's what this is right here, a little outgoing vesicle. Here's your mitochondria. Again, these mitochondria are the power plants. Here's where almost all ATP is made. Got to have that if you want to power these cells, if you want them to live. And then here's some different vesicles. Again, you can't really tell them apart by looking at them. But again, if you knew a little about their function and what they were for and what was to the inside of them, you'd be able to tell them apart. Maybe they have digestive enzymes. Maybe they got something else. Then here's your little centrioles over here to the side. Again, this is where you'll see the spindle fibers forming around them. It'll also make an extra set when a cell gets ready to go into mitosis. This have a lot to do with moving things whenever the cell divides. And of course the cytoplasm, just be all this region in between all these organelles and structures here. Mostly fluid, but there's some other materials in there too. So here's another little picture of a cell model which you can look at. There's little connections to the books on Amazon.